Welcome to this lecture on the exogenous antigen processing pathway. Today, we are going to explore one of the most fascinating and crucial defense mechanisms in the human body. If you have ever wondered how your immune system knows exactly which bacteria to fight, or how vaccines actually train your body to recognize future threats, you are in the right place. We will build your understanding from the ground up, starting with a simple analogy, moving through the cellular biology, and even looking at the mathematical kinetics that govern these microscopic interactions. Let us dive in. To understand how this pathway works, let us start with an intuitive analogy. Imagine your body is a highly secure fortress. Outside the walls, elite guards are constantly patrolling. When they spot a suspicious intruder, they do not just sound a generic alarm. Instead, they capture the intruder, drag them into a secure processing room, and carefully dismantle their equipment. The guard then takes a highly recognizable piece of the intruder's gear, like a unique helmet, and mounts it on a special display board outside the fortress doors. This display board tells the elite tactical SWAT team exactly who to look for and destroy in the surrounding area. This is exactly what happens at the cellular level. Now that we have our fortress analogy in mind, let us look at the formal biological definition. The exogenous antigen processing pathway is a specialized cellular mechanism. It is primarily used by what we call professional antigen-presenting cells, or APCs. The main players here are macrophages, dendritic cells, and B cells, which act as our elite guards. Their primary job is to take up extracellular antigens. These are pathogens or foreign proteins that are floating around outside the cell, in your blood or tissue fluids. The cells capture these invaders through processes known as phagocytosis or endocytosis, effectively swallowing the threat whole to bring it inside for processing. Once the antigen-presenting cell has swallowed the invader, what happens next? Inside the cell, these captured antigens are trapped in secure membrane-bound vesicles called endosomes. Think of this as the secure processing room from our analogy. These endosomes eventually fuse with lysosomes, which are cellular compartments filled with harsh acids and destructive digestive enzymes. When they merge, these enzymes go to work, cleaving the large, complex foreign proteins into smaller, manageable peptide fragments. The cell is essentially dismantling the bacteria into tiny, recognizable pieces that can later be used as biological identification tags. While the invader is being dismantled, the cell is simultaneously preparing the display board. In the endoplasmic reticulum, the cell manufactures major histocompatibility complex class II molecules, commonly known as MHC class II. These molecules are routed to meet the newly created peptide fragments. The MHC class II molecule binds tightly to a peptide fragment, forming a complex. This complex then travels all the way to the cell surface, where it is displayed for the outside world to see. Specifically, it is presented to CD4 positive helper T cells. These T cells are the tactical SWAT team commanders. Once they see this displayed antigen, they initiate a massive targeted immune response. Understanding this presentation process is absolutely critical because it serves as the primary communication bridge between your innate and adaptive immune systems. Without this specific pathway, your body would have no way to alert helper T cells about dangerous infections in your tissue fluids. The helper T cells must physically see these presented peptides to authorize B cells to produce targeted antibodies. Furthermore, in real-world medicine, this pathway is the exact biological mechanism that makes modern vaccines work so effectively. When you receive a vaccine, your antigen-presenting cells take up the harmless vaccine particles via this exogenous pathway and display them, perfectly training your immune system for future real-world encounters. Now that we understand the biology, let us look at the underlying chemistry. The physical interaction between the peptide fragment and the MHC class II molecule is governed by strict chemical kinetics. We quantify this using the dissociation constant formula. Conceptually, this formula tells us how effectively a pathogen will be displayed. In this equation, Kd represents the dissociation constant, which measures how easily the complex falls apart. P is the concentration of free peptide fragments, and MHC is the concentration of empty MHC molecules available for binding. Finally, the P-MHC complex represents the concentration of the successfully formed pairs. A lower Kd value means the peptide binds very tightly, which is ideal for triggering a strong, long-lasting immune response. If free peptides increase during a heavy infection, more complexes will naturally form. Let us apply this formula to a practical biological scenario. We are given a free peptide concentration of 1.5 times 10 to the negative 6 molar and an available empty MHC concentration of 2.0 times 10 to the negative 6 molar. The dissociation constant is 3.0 times 10 to the negative 7 molar. We need to find the final concentration of the formed complexes. First, we rearrange our formula algebraically to isolate the complex concentration on the left side. Next, we substitute our known values into the numerator and denominator. Multiplying the top gives us 3.0 times 10 to the negative 12. Dividing this by our dissociation constant yields a final answer of 1.0 times 10 to the negative 5 molar. Physically, this high concentration means the dendritic cell is extremely effective at processing this specific bacterial peptide, guaranteeing a robust activation of helper T cells. Once these peptide MHC complexes are formed inside the cell, they must be physically transported to the cell surface. This brings us to our next mathematical model, which calculates the biological delay before an immune response can begin. We model this as a linear growth problem. The formula states that the threshold number of complexes equals the initial baseline number, plus the rate of transport multiplied by time. N threshold is the target number needed to activate a T cell. N0 is the starting baseline of complexes already present. R is the cell's internal logistic speed, or how many complexes it moves per minute. Finally, T is the time in minutes. If the transport rate increases, the time required to reach the threshold will decrease, leading to a faster immune response. Let us see this linear growth model in action. Suppose a passing helper T cell requires a minimum threshold of 4,000 complexes to become activated. The macrophage already has an initial baseline of 500 complexes, and it transports new ones at a steady rate of 50 complexes per minute. We want to find out how long it will take to reach the activation threshold. We start by substituting our known values into the linear equation. Next, we subtract the initial 500 complexes from the 4,000 target, which tells us the cell needs to transport 3,500 additional complexes. Dividing this net requirement by the transport rate of 50 gives us our final answer, 70 minutes. This calculated time delay perfectly explains why it always takes a noticeable amount of time for our immune system to ramp up its defensive response after encountering a new pathogen. Now that we have calculated the kinetics and timing, let us look at the entire process visually. In this diagram, you are seeing a professional antigen-presenting cell. Starting on the far left, Notice the exogenous antigen being engulfed into a vesicle called a phagosome. As you follow the arrows inward, this phagosome fuses with a lysosome, creating a phagolysosome, where the antigen is digested into small peptides. Meanwhile, look at the top right. The endoplasmic reticulum is synthesizing the MHC class II molecule. This empty display board travels down to meet the peptides. They bind together, forming the final peptide MHC complex. Finally, follow the pathway to the bottom right, where the complex is pushed to the cell's surface. There, you can see the CD4 positive T cell physically docking with the complex, officially triggering the adaptive immune response.
We have covered a lot of ground today, so let us summarize the most important ideas. First, remember that professional antigen-presenting cells act as our fortress guards. They engulf extracellular threats and digest them into small, manageable peptides. Second, these peptides are carefully loaded onto MHC class II molecules, which act as biological wanted posters. Third, these loaded complexes are transported and displayed on the cell surface, specifically for CD4 positive helper T cells to inspect. Finally, this entire presentation mechanism is absolutely essential for activating the adaptive immune response, driving antibody production, and ensuring long-term immune memory, which is exactly how vaccines protect us. You now have a comprehensive understanding of the exogenous antigen processing pathway.